So I'm Ben Raphael. I'm in the computer science department at Princeton University. This is actually my fourth CGSI, and I was sort of commenting on the break, sort of lamenting this move to the new room, which is, sort of corporate and not like the old 80s, style room we had back at the faculty club a few years ago. But then, the screen is not really working, so it gives us that nice renegade vibe that we're used to at CGSI. A little randomness in what you're going to see on my slides. And I didn't have time to readjust my slides for the gap there, so we'll see what we can see. About 2 months ago, they asked me for a title, and I gave them some title like this, but, frankly, I never know what I'm going to talk about until the day before. So I will talk about this, so it's not truly false advertising. But I'll have a bonus topic. So if you don't like cancer and DNA, I'll talk about RNA and no cancer in the second part. Okay, so a quick disclosure slide that they require me to have. And I think the setup here has been very well set up by Quaid this morning. Cancer, if you didn't know before, now you know from Quaid's talk, is driven largely by somatic mutations, both passenger mutations that are just background mutations, and then driver mutations that give the cells containing them some selective advantage. And in the past decade or so, we've now been able to sequence tumors and measure these mutations, and we find that there are mutations across all genomic scales, from single nucleotide mutations all the way through larger copy number aberrations and structural rearrangements and things like whole genome duplications. And then the other thing you learned from Quaid's talk this morning is that cancer is also an evolutionary process. These mutations that we observe at the present time are the result of these accumulation of mutations and selection for advantageous mutations. And this process is ongoing in the tumor sort of naturally, but also in response to various treatments. So one of the things that has been of interest in the past couple of years is to try to take the rich genome sequencing data we're obtaining from cancer genomes and try to reconstruct the past and figure out what was the past history of this tumor. And this is a problem that's a well-known methods problem, a well-known computational problem of phylogenetic tree reconstruction. And we can think of building a phylogenetic tree that represents the tumor phylogeny. And we'll hope to sort of obtain these major clones that's underwent these clonal expansions and obtain a tree that shows these clones on the nodes, the ancestral clones, the current clones, and then the mutations that distinguish parental clones from their children. And so why might we like to do this? Well, there's many reasons. I mean, other than just the intrinsic interest of doing this, by having a better notion of the tumor and what it looks like at the present, but what happened in the past, it might give us a better understanding what the drivers were of the cancer in that individual patient. If we do this same sort of analysis on longitudinal samples, we can in some ways identify what might have been the mutations that are responsible for a response or a resistance to treatment. And if we sort of do the same type of analysis from a primary tumor and metastases from the same patient, we might be able to reconstruct how and when did those metastases sort of migrate from one anatomical location to the other. And this might then help us inform what might be the driver events of metastasis. So, the standard approach, right, what you learn if you take a basic bioinformatics class or an evolutionary biology class is that, here we would have sequenced and detected somatic mutations in the cells of this tumor. I can represent these in a simple way as each somatic mutation either being present or not in individual cells. And then I go, run one of the many sort of standard algorithms for doing this. And there's many, and there's, many books and volumes written on the different algorithms and when they're useful and how to estimate robustness, et cetera, right? So what's sort of new and different here? Well, as was alluded to this morning, I mean, one thing that's different is that actually most of the sequence that we've done is of bulk tumor samples. So we don't sequence individual cells, we sequence a tumor mass, maybe hundreds of tens to hundreds of thousands of cells. And what we then obtain is not a mutation present or not in individual cells, but we obtain this sort of notion of the frequencies of mutations in this population of cells. And the way we get estimates of those mutation frequencies is by aligning the reads to the reference genome, identifying somatic mutations. Here I'm showing a single nucleotide mutation, and then from the fraction of reads that align to this locus that have the somatic mutation, that gives us an estimate of the so-called varying allele frequency. And this varying allele frequency is sort of proportional to the population frequency of the mutation. So that instead of having a traditional phylogeny problem, a sort of max parsimony or max likelihood problem from data like this, we instead sort of have observed superpositions of signal, right? We've observed superpositions of the rows of this matrix in terms of these frequencies. And so this leads to then a whole host of interesting problems, which you could think of as sort of phylogenetic deconvolution problems, where you're sort of separating out this mixed signal at the same time you're trying to reconstruct the tree. And this has been a very active area in computational biology in the past few years. Sort of few of the many references here, a couple of Quaid's here, a couple of R's. I put ours in bold since I'm giving the talk. And these are all sort of for generic multi-sample sequencing. But actually, if you start making use of other structure in the data, like if your samples come from different time points, longitudinal data, you can actually make use of that information when you do this deconvolution. And we published an algorithm a few months ago called Calder. Matt Myers is the first author who's here at CGSI. You could talk to him about this. And then if you make use of information from different anatomical sites of the same individual, from primary tumors and metastasis, you also get additional information that you can use in this deconvolution problem. And we published an algorithm called Machina last year that I presented at CGSI last year. So you can go watch the video and learn about that. But the question I get most of the time when I present these things, especially these days, is, well, we can do single cell sequencing now. So you don't need to do all this fancy deconvolution stuff, even though it's really cool algorithmically. And actually, I should say there's a bunch of unsolved problems here that still are useful. But, just go sequence single cells. And often when I get this question, it's because someone's been dazzled by seeing some slide like this, where the number of cells that you can sequence from droplets is sort of going up in this exponential or super exponential way, or they've gone to a bunch of talks where they've been dazzled by dozens of t-SNE plots, and first in answering this question, the first thing I realized is actually a very simple thing that I think all of you at CGSI know, but I'll just sort of reiterate this, which is that DNA is not RNA. So the fact that we can do single-cell RNA sequencing doesn't mean we can actually do single-cell DNA sequencing. It's sort of depressing for me because I realized, wow, I just gave this whole talk about DNA, and then this audience member didn't even realize that I was talking about DNA and not RNA the whole time. So a lot of the cool stuff you see is single-cell RNA sequencing. And there are various reasons for this. And if you try to do this with DNA, you find out that there's missing data and there's lots of errors because you have to amplify the single molecule of DNA, and you sort of have the sampling issues. And also just for these practical reasons and also for historical reasons, the vast majority of cancer sequencing data we have is from bulk tumors. People aren't doing single cell sequencing clinically, so these algorithms are still sort of useful. But in the past year, I've kind of come around to the fact that maybe I've just been being too conservative about this whole thing. And single cell sequencing really in many ways is sort of a technology that's really in tune with modern times, because although for each individual cell that we sequence, we get really low quality information. We don't get sort of the quality of information that we get from bulk sequencing data. But just as these days we sort of get our news or our friends or our dating prospects, not from a few high quality individuals, but by surveying thousands of low quality individuals and hoping that we can somehow separate the signal from the noise. Why not just do the same thing with our sequencing, right? Maybe one of these cells we sequence is garbage, or maybe they're all sort of undersampled, and maybe Russian bots have contaminated the whole data set. But let's just go with it and see what we can do. So for cancer, we can think about a very sort of simple crack at this first, which is let's try to sequence these single nucleotide variants. Okay, and as I said, we can extract single cells. We can actually do this, using fancy microfluidics now. But there's only one molecule of DNA per cell, so that's not enough signal to sequence. And so you have to do some clever tricks with whole genome amplification in order to get enough DNA to sequence. And then after those tricks, you can go do targeted sequencing of individual genes, or whole exome sequencing, or whole genome sequencing. But we don't get this beautiful matrix of cells by mutation, zeros and ones, because when we do this whole genome amplification step, there are various ways to do this. Sort of a nice review article describing some of the ways that people have developed to do whole genome amplification, but they're all sort of some variation on PCR, really. And so when we do this, what we end up with is this sort of uneven coverage per cell, which leads to things like, we may or may not detect, we may or may not measure the allele containing a somatic mutation in any cell that we sequence. We may have no reads at that locus. We may only have the reference reads and not the variant reads. And so, instead of getting a 1 here, we get a 0, or we get a question mark because we have no data. Or, when we do PCR-based sequencing, if we had any sequencing error in early steps, that's just going to be amplified. So we also have sort of, false positive zeros that became ones. So, doing single cell sequencing doesn't obviate the need for computation, which is good for those of us in the room. And in fact, it opens up a whole host of interesting algorithmic problems. And these really we can think of, as sort of phylogenetic imputation problems, where we've got now this matrix. It's, at the single cell level, but instead of being the nice 0, 1 value matrix, it's, got a lot of errors. We got some question marks, we got some ones that should have been zeros that should have been ones. And so can we recover the true signal? And of course, we can attempt to use the fact that there's, some relationship between the cells and use something like phylogenetic approaches. So I'm going to show one method that we've developed recently for doing this. There's work by my PhD student Greta Satas. It's a method called Scarlet. Okay, so the basic first observation here, right, is that this mutation matrix has missing entries and it has errors. But the matrix isn't random. It's the rows of this matrix, the cells, are somehow correlated by the ancestral relationships between the cells. So there's information that we should be able to use in order to help us impute what the true matrix should be. So in order to do this, we need to really have some model for what the evolutionary process should be. And the structure that we have in the matrix is intimately linked with the model that we use. And so the early work in this area focused on what is the simplest phylogenetic model, which is based on something called the infinite sites assumption, or the no homoplasia assumption, which is to say that if we think of 0 as the ancestral state, the sort of unmutated state, the normal cell, then over this evolutionary process, each mutation will change from 0 to 1 at most once. Okay, so this means each edge of the tree, when I indicate this mutation, that's it changing from the 0 unmutated state to the 1 state. And this infinite sites model in the computer science literature is actually sometimes also called the perfect phylogeny model. Here I'm sort of specifying the ancestor as being all zeros, and it's actually a very simple and straightforward model to work with. So given this mutation matrix M, the perfect phylogeny is the rooted tree, the leaf is all 0. Every mutation, because it occurs only once, labels exactly one edge of the tree. There's a unique path from the root to the leaf. That's true for any tree, but then you can just trace out what are the mutations that occurred on that path, and those are exactly the ones you should have in the row of the matrix. And you could compute this in time that's linear in the matrix, and the matrix M that satisfies perfect phylogeny has a very special structure that's given by the 4 gametes condition. I won't go into sort of what exactly that is, but there's many good references here. Dan Gusfield at UC Davis has done a lot of the seminal work in the computer science community to work these things out. Okay, so we don't measure that structured 0-1 matrix, right? We measure this matrix with errors. And so what we'd like to do is, if we assume that the process that we observed is a result of an evolutionary process under this infinite sites model, we'd like to make the sort of minimum number of edits, the minimum number of flips where zeros flip to ones or ones flip to zeros, or if we have a likelihood model, maybe the most likely sequence of flips to get back a perfect phylogeny matrix. Okay, and so doing this, if you actually try to compute the minimum, turns out to be a computationally hard problem. It's a so-called NP-hard problem. So what's been done in the methods that have been applied to cancer sequencing data is to use a more probabilistic approach, which is also valuable because you can model the probabilities of individual errors, and then you try to compute the probability of a given tree, probability of a tree given the data by Bayes' rule and MCMC, for example. Okay, so the sort of seminal method in this line was a method called CITE by Nico Baron-Winkel's group, and this is what sort of motivated our work in this area that I'll now describe. Because we saw this paper from Nick Navin's group where they had sequenced a primary colon cancer patient and metastasis from the liver. They'd done single-cell whole exome sequencing, and they ran this CITE algorithm and built this tree based on the single nucleotide mutations. And the cool thing about this tree and about this paper was what that they observed was that from the primary colon tumor, as you trace through the mutations, there was 2 separate migrations from the colon to the liver met. So this is something called polyclonal seeding of the liver met. Something we sort of worked on in the bulk sequencing case. That was the Machina paper that I mentioned 10 slides ago. So we thought, oh, this is really cool because now we can sort of see these types of things in single cell data. But as we looked further into it, there's a little bit of strangeness in this result. Because one thing that hadn't really been done here was to look at the other types of mutations that also were occurring in this patient. And in particular, there are many, many copy number aberrations, right? So there's mutations across all scales. We can build a tree by choosing one class of mutations, but really we should be thinking holistically. And copy number aberrations are very common in solid tumors. If you look at colorectal tumors, this is like TCGA data. Each column here is a patient. Across all different chromosomes, you see the normal 2 copy is diploid. You see lots of blue and red, which are duplications and deletions, over the whole genome in almost every colorectal patient, cancer patient, TCGA. Okay, so we can of course detect these copy number aberrations, by also aligning reads to the genome, and even with single cell data, and we can look for changes in the read depth. So that gives us some notion of whether there have been duplications or deletions at certain locations. And so we did this, and actually the original authors had done this as well for the same colorectal cancer patient. So here what we're seeing is the individual cells here are colored by whether they were from the primary tumor or the met, and across the genome, the copy number profile. Okay, so the colors here indicate copy number changes. White again is the normal diploid state 2, and what you can see is that there's dramatic changes in copy number. Okay, but also what you can see is that, here's the primary cells in blue, here's the met that's sort of split off. The metastases cells have very different copy number profiles, yet somehow what happened on this tree is magically the second met obtained almost the exact same copy number profile. So it's like the genome knew to completely remodel and change its genome dramatically 2 separate times in order to see this metastasis.
Have you given this talk to Nick Navin?
Not yet, no. He'll see it on video. So this is pretty remarkable, but also like a little bit suspicious, right? So what's perhaps going on? Well, we have to think about both single nucleotide variants and copy number variants accumulating on this tree, right? And interestingly, they don't, they aren't independent. In fact, there's a very strong way in which copy number aberrations and single nucleotide variants interact with each other. Because if you have a somatic mutation that is in a region that's later deleted, that deletion can remove the mutation. Right? So it's a very strong interaction between these 2 types of mutations. And so we have to be very careful of building trees on single nucleotide mutations and the assumptions we're making, because mutations can be lost due to these copy number changes. So what this really points to is this infinite sites model, if you think about copy number changes going on, is much too simplistic. Right? 0 going to 1 only once, because this 1 could later be deleted, and then it goes back to 0. Right? So it wasn't a back mutation in the single nucleotide sense, it was a loss. Okay? And it turns out these are actually quite common, and the the authors of CITE, and I guess they included me along here for various reasons I can describe, had sort of seen that this infinite sites model is not really strong enough. So we have to relax the model. Okay, so we can relax the model in various ways. The most simplest relaxation is to say, well, we really don't think that a mutation, a single nucleotide mutation, occurring probably only occurs once, but maybe it can be lost many, many times over the course of the tree because of copy number aberrations. So this idea of 0 going to 1 once, but being lost many times is so-called Dolo model. This can be computed, or we could just go sort of to the most general case and allow mutation to go from 0 to 1 back to 0 as many times as we need on the tree to explain the data. This is just the general purpose so-called finite sites model that's used in evolution. All right, so it turns out it's a little bit harder to compute this, but that's not really actually the issue. Is that what really becomes the issue is as we relax the model, there actually becomes lots more trees that are consistent with the data, and there's a lot of more ambiguity in our data. And you can see this just in sort of a simple case. Here's just 3 cells with these individual mutations. If we allow loss, I can explain these 3 cells in sort of 2 different ways, where there's each mutation happening only once, but between the 2 trees, one or the other mutations is lost. And as you sort of make this bigger, there just becomes an explosion of the possibilities that you can obtain. So what we sort of realize is well, there's no reason why we have to think of just mutations as these individual locations, and maybe they're gained or lost, and it's because of copy number aberrations, because we know exactly where the copy number aberrations are. So we can actually use the information about these copy number profiles in the individual cells to help inform where we should expect these mutation losses to happen. And if we don't see a loss in copy number at a certain locus, here we have the same copy number profile between these 2 cells, we wouldn't expect a mutation to be lost there, compared to the case where there's a deletion between the 2 parental cells right at the location of this single nucleotide mutation. So we want to use this information that we have in the copy number profiles to constrain the model. So what we can then do using copy number aberrations is define what we call supported mutation losses. So these are your loss of a mutation, one going to 0, but only on edges of the tree where there is a copy number change, a decrease in copy number at that location. Okay, so this leads to sort of an intermediate model. You can think of this as this Dolo model where 0 goes to 1 only once, but there can be many losses. But now we allow losses, but only at locations where the copy number data tells us there could possibly be losses. Okay, so this is a loss-supported model, so it combines thinking about both copy number and single cell mutations sort of jointly. And then now to actually turn this into something practical, we have to think about the specific characteristics of the data that we have. And it turns out that with current single sequencing technologies, DNA sequencing technologies, you can sort of get single nucleotide mutations measured with high confidence, but then you don't measure copy number with high confidence. Or you can get copy number measured with high confidence through some tricks, but then you actually don't get single nucleotide mutations measured with high confidence. So there's sort of a trade-off. And so the data that I showed you from the colorectal tumor, actually because they did this targeted whole exome sequencing, really was in the regime where they had very good information about single nucleotide mutations, but not really very good information about copy number aberrations. So that means that the copy number profiles we obtain are going to be, sort of a little more global, and we're not going to, really be able to identify small changes. So what it means is that if we build a tree of the cells based on only their copy number changes, it'll be really a small number of copy number profiles. The tree will not be very well resolved. Okay, so we can sort of assign a cell to one of a small number of copy number states. Okay, so that's the regime of the problem that we worked out. So then we sort of start with this copy number tree, which is really only derived in a small number of copy number states. So actually we can build this tree efficiently because there aren't really that many copy number profiles that we obtain. And then we take the single nucleotide data and we use it to refine this tree. And by refining this tree, what I mean is that we resolve some of these multifurcations and resolve them in a way that nodes here correspond to subtrees over here. Okay, and we do this in a way that preserves the sort of infinite sites model on single nucleotide mutations, but allows losses on edges where copy number state changes. Okay, and we can actually, and this is sort of the fun part of, I guess, working in sort of methods, here's sort of a different problem, so to speak, but actually the algorithm for solving this problem reduces to something called the incomplete directed perfect phylogeny problem, which I'm not going to tell you, except to say that it was worked out by Itzik Pear, who I think was at CGSI in the first week of the program here, right? So here we can pull a 15-year-old algorithm off the shelf and with some changes actually use it for a sort of new problem. So hopefully in CGSI 2034, someone will be doing this with one of my algorithms. I can only dream. So that's not the whole thing, because actually there are these different classes of errors, and we want to really model the copy number counts reliably and model the different rates in these errors. So, we actually sort of do a probabilistic model of the sequencing data, whether there's a 0 or 1 or question mark, and we actually compute sort of a maximum probability version of this incomplete directed perfect phylogeny. And we solve it using actually a combinatorial approach in ILP. And this leads to a method that, which is acronymed as Scarlet. So that was actually quite an accomplishment of Greta to figure out a cool name for the algorithm that involves single cell, right? We do some benchmarking, right? We simulate data with losses. If you use the infinite sites model, of course, you're not modeling losses, so you don't expect to really be able to get the tree reliably, and of course, that's what we see. But kind of interestingly, even if you use these models, and you can't even see this says Dolo model. I should look back at this to know what you guys aren't seeing, because I'm looking at the screen that looks at everything. So even if you allow losses with the Dolo model or with this finite sites model, actually you still sort of do not that much better than not allowing losses. And we think a lot of this just has to do with the ambiguity. Like there are just so many ways you can have losses. And if you don't sort of look at the copy number aberrations, you really just end up producing solutions that have maximum likelihood or minimum number of changes according to these models, but the losses are all in the wrong place. Right? And same with sort of the tree error, and we can see that we have the sort of the smallest tree error. Let's go back and look at this data set, right? So we're able to, take the single nucleotide data that was published, derive copy number profiles. We really don't need the whole copy number profile. All we really need to do is for each mutation be able to say, whether there's a change in copy number that would support a loss. And so that's sort of what goes into the loss supported model. And so then when we go back and look, here's again what was published via site. We have this polyclonal seeding. And then if you look at where losses are allowed, we find that we can build a tree where each mutation is acquired only once, but there are 3 individual losses on the tree that lead to actually a much more consistent phylogeny, where you have the same copy number state, it changes only once, and that's the change that is the copy number state in the metastasis. And what this also then leads to is the fact that there's monoclonal seeding. So we'd only infer a single migration from the primary tumor to the liver mets. And each of these, so you can see the copy number changes, each of these mutations, we see concomitant copy number change. All right, so that's the summary of the first part. Lots of ambiguity in single-cell DNA sequencing data, but here the phylogenetic tree provides some structure that we can use to inform that. If we're looking at cancer evolution, we have to be aware and we have to be careful that there's mutations across all different genomic scales, and so if we're not careful about that, we can end up with sort of strange solutions. And so Scarlet is sort of one way of doing this that integrates both single nucleotide mutations and copy number changes. And at least on this data set, we see that maybe this idea of polyclonal seeding is not really so well supported. Yeah. How big are these? This is, so what I'm not showing here, there's a collapsed version. There were, a few hundred cells, 186 cells. So what you're seeing here is a collapsed representation where multiple cells are connected to each of these nodes. But this is the, other than that collapsing, this is the whole tree.
Why only 186?
Because it was this early technology that was based on doing these early versions of microfluidics. I forget if it was Fluidigm or what it was, and they do this whole genome
Ran the
amplification.
algorithm on thousands of, tens of thousands of cells.
We could do that. There might be a couple, scalability issues with the current algorithm. It would go to 1,000 probably. Whether it would go to 10,000, I'm not sure. Fortunately, for us, at least in terms of the algorithm, no one has, single cell whole exome data from 10,000 cancer cells yet from the same patient. It's sort of these trade-offs, but there are cool technologies. 10x Genomics has this single cell kit for copy number variation. So they do low coverage whole genome sequencing using their technology, so you can get 1,000, 2,000 cells on a single run of it. But you're not getting, you're getting like 0.0004x coverage per cell, so you can't really get single nucleotide mutations. We can get copy number profiles. So. Okay, so now something sort of different in the last few minutes here, and since this is CGSI and there's a lot of trainees, I thought I would sort of tell the origin story of this project because it is different, and the origin story is really one of my grad students, Rebecca Eleno, saying, I want to work on single cell RNA-seq data because it's cool and there's lots of people doing cool stuff and there's t-SNE plots everywhere. And I sort of said, well, we're kind of a DNA group, not really RNA. RNA is messy, it changes all the time. DNA is pretty stable. These arguments weren't working. I said, and all the RNA-seq people fight with each other all the time and argue, and maybe we don't want to get involved in that. It's pretty competitive, but none of my arguments worked. And so I said, okay, fine. Well, let's try to find an angle here that might be interesting. And one of the angles was to actually think about something that I know some of you in this audience really love. So think about some of our earlier work, which is on networks. So I said, let's do something with, maybe we can use networks and single-cell RNA-seq. And this led to sort of a method that has this not as great name, MMF SC, but let me tell you a little bit about that.
You didn't use HotNet 3?
No, we couldn't do that. You'll see why in a second. So single-cell RNA-seq, hey, it's really cool despite my, I'm joking a lot about single-cell, but obviously I'm presenting it, so, it is a cool technology. With the droplet-based technologies, specifically, what you end up getting is you can sequence lots of cells, but with each cell, you really undersample. So you get lots of zeros in this sort of matrix of transcripts by genes, whether or not you measure the transcript in individual cells. And actually, with the 10x technology, these dropout rates, the number of zero entries in this matrix, it can approach like 90-95% in some data sets. Okay, so what this means is if you try to look for structure in this really sparse matrix in a naive way, like doing PCA, you quickly find that the largest source of variation in the data is the dropout rate, right? So here's just the values of the first principal component as a function of the zero fraction in that cell. This is from a few years ago, but still true, even more so, I think, in some of the current data. So what can we do? Well, we can combine information across cells, right? I mean, we've measured many cells, and they're not all distinct, right? There's groups of cells that are in the same cell type or cell state, and so if we take this matrix and maybe compute some cell-cell similarity matrix, then we can help overcome this problem. Of course, there's a lot of challenges in this because even computing that cell-cell similarity, the dropout is actually still there, so it becomes actually the dominant signal in the cell similarity, and you sometimes have to do some tricks even to get there, which is what some of these methods are sort of doing. And then another approach is to say, well, let me only look at highly expressed genes. I reduce to a small number of genes ahead of time before I do any of these things, but this can result in some amount of bias. So the other thing, of course, you can think about is the fact that, well, not only are cells not all the same, but across different cells, there are groups of genes that should be sort of similarly expressed, right? Because genes are, in pathways or protein complexes. There are all sorts of co-expression we might expect from individual genes. So maybe we can now think about taking this matrix and, sort of grouping it into groups of cells that have similar expression and groups of genes that have similar expression across cells, right? And if we sort of take what I sort of wrote down there in a very specific way, we could sort of think if the dimensionality of these two things is the same, we could, do something like non-negative matrix factorization. We could just factor this matrix X into, a gene by cells matrix and a sort of latent dimension by genes matrix and the same latent dimension by cells matrix, and that might give us a sort of low rank approximation of the data. And that's a good idea, but actually sort of doing this naively doesn't really kind of address the dropout problem directly. And so people have, again, tried to maybe limit this to highly expressed genes. So what we sort of thought about is, well, could we use some sort of prior knowledge to help guide which genes we think might be similarly expressed across cells? And at first we were thinking along the hotnet lines of using protein-protein interaction networks or some sort of silliness like that. But then we realized, wait a second, people have been doing RNA-seq for a long time, and before that they were doing microarrays. And one of the fundamental things that they always derived were gene-gene co-expression networks, right? So there's tons of database. I mean, there's huge databases of thousands of RNA-seq and microRNA derived gene-gene correlation networks, right? So this actually gives us a notion of genes that should be co-expressed across, in fact, different tissue types, sub-tissue types. I mean, there's really a huge, rich data source here. So what we sort of thought was like, let's use this co-expression network to guide our factorization of this matrix. And we can just extend NMF with a sort of regularization term where what we try to do is in this gene embedding, we sort of try to make genes close in the embedding if they were co-expressed in the network. Okay, so using sort of this network, we can write it down as a little Laplacian, and this has all been sort of worked out in other applications, something called graph regularized NMF. Okay, so we thought let's sort of do graph regularized NMF. And there were a couple, sort of small tricks. In fact, there's two types of NMF. One is in the Euclidean distance. One is in the KL divergence. This actually turns out to sort of model a Gaussian loss function, whereas this one models Poisson. And there's some, tricks with how we do the correlation, but all really small tricks. This is essentially this graph regularized NMF. And the results I'll show you are from the KL divergence sort of version. Okay, so that's really it. We take the sparse transcript count matrix, some gene-gene co-expression network, and I'll show you which ones in a second. We run this network regularized NMF, and what we hope to get out is here's the product matrix WH. The individual matrix H gives us a clustering on cells, so we can look for cell states or cell types this way. We didn't use deep learning, but we want it to be sort of cool, so at least we use TensorFlow to do the optimization. So it's all implemented in TensorFlow and on GPUs. It was a tough crowd actually for the sort of jokes, it runs pretty fast. Okay, so what do you first do? You check how it works on simulated data. Here the simulated data is we take one of these gene-gene co-expression networks. We sort of derive clusters from the data. We then subsample it. So I should say and then the network we use for net MMF SC is the original sort of larger gene-gene co-expression network. So it's not the exact same correlations that you expect in the sparse data, but of course it's related. And unsurprisingly, because we've kind of cooked up the example, by using the network information, we do much better in recovering the 6 clusters than these other sort of competing imputation methods. Okay, so that's just sort of a proof of concept. Now when we go to various single-cell data sets, we see how we do. So this was an earlier data set that had 182 cells. They were flow sorted by cell cycle so that we have some notion of the true cell cycle state of every cell. So that's sort of the gold standard. And then we try to cluster cells by their cell state, and we find that we do the best at recovering the true cell states than many of the other competing methods. Interestingly, this method MAGIC, it's really sort of a trajectory method, not a kind of finding cell types method. So it infers this nice trajectory here, but it's not really necessarily representative of the cell type clusters in the data set. What about the network itself? Well, if we change the network that we use in NetMMF, it of course, changes the performance. That's good. And what we found is that, the more matched of the co-expression network we use, the better performance we obtain. So if we're analyzing, mouse embryonic stem cells, as we sort of were in this case, we get the best performance if we use a co-expression network derived from mouse embryonic stem cells, which conveniently is sitting in a database somewhere. And that's better than using just a generic mouse expression network. So that's sort of nice. And if we randomize the edges of the network, actually we get back the performance of NMF, which is also sort of a good reasonable thing to have happen. What about in a larger data set? Here is brain cell types from 10x Genomics. This is a very big data set, 1.3 million cells. And then they had a smaller version of this data set from a different mouse with only 2,000 cells. So what we tried to show here is that we could recover the cell type structure using the small data set. So we use the big data set as the gold standard. Comparing against other methods, what we sort of saw was that our approach to MMF SC here actually sort of matched the population proportions of the different cell types well compared to other methods, which, for example, had large fractions of cells that were unclassified, that didn't match to any of the particular types, or had frequencies that were very, very different. Sorry, these are the glutamagetic neurons or something, right? So we match the large data set almost perfectly, and a lot of these other methods are sort of wildly off in the population proportion of these different cells, 21%, 39%, et cetera. Okay, so what we sort of saw is that, yeah, we matched these proportions of these cells sort of the best using, several orders of magnitude fewer cells. This sort of shows how, using the structure in the data as informed by this brain cell network from mouse allows us to actually recover these cell types with many, many fewer cells than was used here. And many of the proportions, almost all of them, are actually within 2% of the proportions found in this other cell type. I think I've probably chewed up all my time. So let me not show you this part about how well we do in recovering gene-gene correlations. I'll sort of skip that part and just wrap up to say what we're doing here. So the approach, again, is to use prior knowledge. In this case, the prior knowledge is in gene-gene co-expression or gene-gene correlation networks to try to help impute and find clusters in single-cell RNA sequencing data set. We focused on the clustering problem, but you could think of extending these ideas to all the other sorts of things that people want to do with single-cell sequencing data, like trajectory inference, find nonlinear manifolds, et cetera. And so sort of generically, what I would say, if we think about new methods in single-cell sequencing, I expect the data is going to be pretty sparse for a while. The paradigm seems to be sequence lots of cells, poorly, because it costs a lot of money to sequence lots of cells well, and then use algorithms to, fill in the missing information, right? So, of course, if we get enough cells, you can just start doing statistics on the whole thing. But until then, maybe you think about low-dimensional structure of the data, latent space representations, or you think of, clever or maybe not so clever, if you didn't like this talk, ways to find structure in the data and use that to impute the data. So, the two structures I used were tumor cells being related by a phylogenetic tree. It was a pretty strong signal in the structure that allowed us to sort of fill in the gaps in the single cell, and then these co-expression networks. So I'll just close by acknowledging that the folks that worked on this. I mentioned Rebecca, but the NetMMF was also was a collaboration with Barbara Engelhardt and her student Bianca at Princeton, and then Greta. Greta was the first part of the talk for the tumor cell phylogeny. And if there's any time, I'm happy to take questions, or we can take them at coffee as you prefer. Thanks.